Hello everyone. For the ones who don't know me, I'm Ms. Ruba Hamouche and I'm a biology teacher at Hoorschwer Public Secondary School. I am glad to present for you the activity for identical reproduction and cell cycle in the chapter 2 DNA, genetic information and cell cycle in the Lebanese program. So let's start our presentation. Do you know that cells need to make a copy of their DNA before dividing? The copying of the DNA molecule is called DNA replication that takes place during interphase which precedes mitosis. Now, why is DNA replication important? Cells replicate their DNA so that the number of chromosomes is conserved from the mother cells to the daughter cells, and so that each daughter cell has a complete copy of this genetic information. As you know, the DNA molecule is the carrier of genetic information and it has the form of a double helix. So each DNA molecule consists of two DNA strands which are complementary and antiparallel. Although mitosis is a cell division, the number of chromosomes is still conserved. How can we explain this? This can be explained by the duplication or the replication or the copying of the amount of DNA during interphase. And this copying of the DNA molecule is done by complementarity of nitrogenous bases. A pairs with T and G pairs with C. So how does the doubling of the quantity of DNA, namely replication, take place? Let's define the DNA replication mechanism. DNA replication is a very important mechanism allowing the duplication or the replication of the genetic information in order to obtain two copies, each transmitted to one of the two daughter cells during cell division. DNA replication therefore takes place before cell division, thus before mitosis during interphase. So chromosomes at the beginning of interphase consist of one chromatid or one DNA molecule. At the end of interphase, each chromosome consists of two sister chromatids or two DNA molecules. The two DNA molecules are formed by DNA replication. Why does this DNA replication take place in the interphase and not directly during mitosis? There are two reasons. The first reason, since during mitosis the nuclear membrane disappears, so the chromosomes undergo condensation, a certain type of compaction, in order to be protected from the enzymes present in the cell which can degrade the chromosomes. Thus, when DNA is condensed, we cannot read it or copy it. In order to be able to copy the DNA, the latter must then be unwound or decondensed during the interphase. The second reason, since during its division, the cell must transmit its genetic information, its whole genetic information, to the two daughter cells, so it is essential that mitosis be prepared by a stage during which all the chromosomes are copied. So that's why DNA replication takes place during interphase and not during mitosis. I gave you here an example of condensed and decondensed forms of chromosomes. On the left, I have represented a folded sheet This folded sheet corresponds to a condensed chromosome observed during mitosis. When the chromosomes are condensed or when the sheet is folded, we are incapable of reading the content of the sheet and copying this content. So, during mitosis, we cannot replicate the DNA. This condensed form is necessary to protect the chromosomes from the degradation by enzymes found in the cytoplasm. Now, on the right, we have represented an unfolded sheet which corresponds to a decondensed chromosome observed during interphase. When the sheet is unfolded, we are capable of reading its content and copying it. So, when the chromosomes are decondensed, we can replicate the DNA forming the chromosome. So, we can say that the chromosomes are permanent structures. They are always present in the nucleus, but they exist in two different forms. The first form is the filamentous or decondensed form observed during interphase. The decondensed form of chromosomes is called chromatin. This latter has a bead on a string form, like a pearl necklace, where the string, the filament here, is the DNA molecule and the pearls are nucleosomes. Nucleosomes consist of one DNA molecule wrapped on four histone proteins. The proteins histone are proteins associated to the DNA molecule and they play an important role in the DNA replication. So during interphase, the chromosomes are in the form of chromatin. The chromatin appears like a bead on a string. The DNA molecule is the string or the filament, and the pearls are the nucleosomes. The nucleosomes contain proteins called histones. Now, when passing from interphase to mitosis, the chromosomes or the chromatin undergoes a condensation. This condensation corresponds to a progressive coiling of the DNA molecule. So the chromosomes are condensed during mitosis, and the maximal condensation takes place during metaphase of mitosis. So at metaphase, the chromosomes are very condensed and well visible. This reaction is reversible. The chromatin alternate during the cell cycle between a condensed form to facilitate chromosome separation and segregation during mitosis and a decondensed form when DNA replicates during interphase. So this reaction is reversible. When passing from interphase to mitosis, the chromatin undergoes a condensation, a progressive coiling. Now, at the end of mitosis, when passing from mitosis to interphase, the chromosomes undergo a decondensation to form the chromatin again. Here, a video that shows you the condensation of the DNA when passing from interphase to mitosis. We have represented the DNA molecule in blue. So this string or this filament corresponds to the DNA molecule, and the red structures are the histones. We have a progressive coiling of the DNA molecule. The DNA is wrapped on the protein histones. So here the DNA molecule undergoes a progressive coiling, a condensation. Here the sister chromatid, and this one is the second sister chromatid. And at the end of interphase and the beginning of mitosis, the chromosomes are made up of two sister chromatids and they are very condensed and well visible. So this is the condensation mechanism of the DNA molecule. Thanks for listening.